All right.
Good. Okay. I was part of the phytoplankton community composition group, but we chose to call ourselves the Bloom, Bloom, Bloom. Our goals kind of changed throughout the project. And so we're going to talk about a comparison of spectra across regions and phytoplankton groups. So because we're all located within the North Atlantic and crossing time zones, we chose to look at several blooms in this region, coastal blooms. So anyway, I'm representing our group from Cassie, Sachi, Devon, and Felix and I. Okay, so the motivation for this group, for this project, was to look at spectral characterization of different blooms. Currently, multispectral imagery that's used for monitoring algal blooms really can only tell you that there's a lot or a little of algae in the water. Most traditional methods are using chlorophyll or fluorescence products to try and find these high biomass blooms. I'm showing on the example from the Chesapeake Bay. This is a chlorophyll A product from the Olchi sensor from the Sentinel-3 satellite. And what you can see is a whole lot of chlorophyll on the western side of Chesapeake Bay, but we can't tell you anything about what's actually blooming there. You know, the microscope is showing what might be in a particular sample in the bay at any one time. So we wanted to investigate whether there are differences in different bloom formers in their spectra by leveraging the PACE data set. So we chose 4 bloom examples in 3 regions, in Massachusetts Bay, Chesapeake Bay, and Florida, the western coast of Florida in the Gulf. So why PACE? It has more hyperspectral bands, and it might be able to give us better indication of what's blooming. This is really important for state monitoring, for fisheries production, is knowing what type of phytoplankton is in the water for their monitoring efforts. Okay, I'm going to pass it off to Felix now.
Hi, everybody. So Florida Fish and Wildlife Conservation, they collect these water samples during the outbreak of red tide all around Florida. Sometimes, as you see on the right side, these red and yellow, these color-coded dots. That shows the intensity of the red tide blooms. So at the peak of the bloom, it can be like 1 million cells per liter. So fortunately, in 2024 and 2025, we didn't have any major bloom that affecting public health in a large scale. But a couple of blooms occurred in fall 2024. So we identified one in 25th of October 2024. So these dots, the white dots along the west coast of Florida, these are the locations where the bloom concentration was more than 30,000 cells per liter. So we have this ABW map for the west coast of Florida, that is from north of Tampa all the way down to the south of Fort Myers. So you can see that red patch that is part of that bloom, and it dissipated if you go further offshore. Can you go to the next slide? So here you can see the chlorophyll concentration associated with that bloom. Again, the bloom is more pronounced between Tampa and Fort Myers. So that is kind of ground zero for the red tide. It usually travels from north to south, corresponding to the prevailing coastal current. There are operational models available by NOAA, I guess collaboration with the USF, predicting the movement of the red tide. So we collected this spectral, the spectra for the selected points where the bloom was pronounced high. And so here you could see there is intense absorption in the blue, and as you go to the 550 in the green area, you can see the peak, and they show the similar trend. And these are all the locations where the bloom was exceptionally high. So they all show the similar trend. for the spectra. Can we go next? This is again the plotting of AVW against the RRS brightness. So you could see that blue color, the intensity of that blue color that is close to the coast, that is aligning very well with the chlorophyll concentration. So that's a clear indication of the bloom. So this is some of the analysis that we learned from this hackathon. So we have this spectra along the transect. There's a transect was drawn across the bloom area and from offshore to nearshore. And on the right side, you can see area normalized spectra. So I would like to explore further on this to see, is there any effect on the abundance and the health of the Carenia brevis bloom as it undergoes through this development, the sustenance, and the dissipation. I would like to explore further in this direction. So this is just only a learning week. So, but I'm glad that I have learned these techniques. So this is a second derivative, and also that is from the, thanks to Ryan's notebooks, very effective, very helpful. So that peaks, I would like to see more how that peaks are affected by the intensity and the health of the Carenia. So that's something that I want to explore further.
Hey, Sachi, I think you're up.
So for the Chesapeake Bay, we had some in situ data for 2 blooms that occurred in July 2025 and August 2024 for Levenderina fissa in 2025 and Heterocapsa species in 2024. And here we have the standard chlorophyll product for Levenderina, and we're not quite sure of what whether these pixels look right. We're kind of debating whether it's atmospheric correction issue or the fact that we are using a standard chlorophyll algorithm for a class 2 water type in the bay. And we have apparent AVW plot and a remote sensing reflectance brightness plots. And I think it's kind of typical to see the AVW is very high at the 2 stations, and we have low RRS brightness that can indicate like absorbing photons, as we learned from the tutorials. And similar with this bloom, we have, this is our station right here, similar pattern, high AVW and relatively low remote sensing brightness. And over to you, Shelly.
Okay, great. So in looking at the spectra at the various stations, because the chlorophyll was really just red everywhere, we chose to look at the ROS data as well, which is the top of atmosphere data. For example, we use the ROS in this chlorophyll product I'm showing here for an Olchi image. So here's our 2 stations just overlaid over the chlorophyll again. And what you can see on the left-hand side is the spectra from RRS and then the spectra from ROS. Keep in mind the magnitudes are different because they're 2 different measurements. And then on the right hand was the similar plots from the 2 stations of the Levenderina bloom in July of 25. And what you can see really, the take-home message is if you're looking at ROS versus RS, there are major differences in the blue. I think that's all due to absorbing aerosols and things that don't get cleaned up because we're using a less corrected product in the ROS. However, if you look from like the green to the red, the spectra look very similar for all of these dates and blooms. Both of these species are dinoflagellates, so I'm not surprised that they look pretty identical. And then we chose to look at a second derivative. So this is all 3 blooms on one plot. Again, the shape of these curves are really, really similar. I will note, I think in the PACE data, there's a gap in some of the wavelengths, unfortunately, so it's not fully hyperspectral in some of these red bands. So we're hoping to get that back in the next level of processing so we can look at the wiggles in that region. And then since we started looking at ROAS, I was able to make an AVW. I have a brightness too that I'm not showing, to look at how a ROAS AVW might look. And I think, again, we're looking at like a lot of absorption going on in the bay, and then a lot of sediment coming out of the Susquehanna River here at the top. I don't see anything that's really indicating our particular blooms in either of these cases. Okay, Massachusetts, I think. Devon, I think you're up.
In the essence of time, I think we're just going to wrap things up here. But if anyone's interested, the slides are available. We did look at Massachusetts using a similar approach. Just to kind of comment quickly, we all have experience using the more standard product of chlorophyll A to look at any kind of phytoplankton bloom. And we're all very eager and excited about looking at phytoplankton blooms using PACE products. If we could just jump to our last slide, that would be great.
Sure.
And we just, agree with all the previous groups that this was an awesome experience. We really enjoyed networking with each other and are looking forward to leaning on those that we interacted with in our group in upcoming weeks, months, years on future projects. And just sharing. Yep, GitHub was certainly quite the experience. Very much looking forward to diving further into these materials, and just the biggest thank you to Eli and Ryan for all the time. Just so inspired to devote oneself that fully to helping others. So thank you both.